the individual and interactive impacts of fire and herbivory on soil microbes. So we know that fire and herbivory influence savanna vegetation above ground, but we know a lot less about what's happening below ground in the soil. So when there's a fire, it removes above ground biomass, and fire has its own direct and indirect impacts on the soil. And then after a fire, herbivores are attracted to the fresh regrowth really quite quickly, and then herbivores also have their own direct and indirect impacts on the soil. And then we have lots of feedback loops going on, all positive and negative, but things like the herbivores are consuming the above ground biomass, so this delays the time until the next fire, and this cycle continues. My PhD is all about understanding this interactive impact on the soil, but I'm just going to talk to you about the soil microbes today. Soil microbes are obviously a really important part of soil health and are involved in key processes in the soil such as nutrient cycling and organic carbon decomposition. So this experiment was here in Kruger, and we used the basalt soils located between Lower Sarby and Croc Bridge. And using a combination of early season prescribed burns and some small exclosures, we managed to get 4 treatments where we have fire with and without herbivory, and then no fire with and without herbivory. And the idea is we can begin to untangle the interactive effects of fire and herbivory. So the fires were burned in May 2021, and they were quite early season and quite low intensity fires. As you can see at the top, they weren't really hot fires. And then the day after the fires, the exclosures were put on the plots, so they're just small 2 meter squared exclosures. And a full year later, in May 2022, we did the soil sampling. So we just had one small experimental low intensity burn and then 1 year of subsequent grazing and herbivory. And then to look at the microbes, I didn't do DNA, I did phospholipid fatty acids, so we get an idea of the size of the microbial community and also the fungal and bacteria counterparts, and also gram-positive and gram-negative bacteria. And we got some really interesting results. So bearing in mind this is just one fire followed by 1 year of herbivore grazing, we found so this is the total size of the microbial community. So on the left we have the fire and herbivory both drivers together, and that has the largest microbial community size. And then when we have fire individually and herbivory individually, we see no differences between the two. And then we also see a decline again when neither driver is present. And this is mainly driven by the fungal community. We see the same pattern with the fungal community here, where we have the largest fungal community when both fire and herbivory is interacting. We didn't see a difference between the treatments in the bacterial community, but because we had seen this shift in the fungal community, we also got a shift in the fungal to bacteria ratio. And what's quite interesting about this is in soil science, we can use a fungal to bacteria ratio to give us an indication of soil carbon storage potential. So this shows that there is potential with the combined interactive effect of fire and herbivory to store more carbon in these basalt soils. We also saw this trend with the gram-positive bacteria, but not with the gram-negative bacteria. And what's interesting about gram-positive bacteria is they tend to be better decomposers of recalcitrant carbon. So the more locked up carbon, that is more difficult for some microbes to access. So this could potentially be as a response to charcoal in the soil after a fire, So individually, with fire and herbivory, we did not observe a difference between the two, but we did see an increased size of microbial community and shifts in composition with this interactive effect. And this suggests that early season burns and the subsequent herbivore grazing could be an important management tool for savanna soil dynamics. This experiment's been going on for 3 years, and we've continued the burns in 2022 and 2023 and did a whole host of soil nutrients and soil respiration, and also some lab-based substrate-induced respiration experiments. So if you're interested, we found lots of other interesting results. So please come talk to me. And thank you to everyone that worked on this project. So Tertia and Sally and Carla, it was a very big collaborative project. Thanks.
Our next talk is by Mary Ngugi. She will be giving us a 5-minute talk on fire and herbivory and how they interactively suppress the survival and regrowth of trees in the savanna.
Okay. I'm Mary. I'm going to be presenting about some work I did for my master's work on fire and herbivory. By now, we know that vegetation structure is the defining feature of savanna ecosystems, and there are drivers that do influence these vegetation structures, and I'll be focusing on the top-down drivers of these vegetation structures, which are fire and herbivory. So in black cotton soil with moderate rainfall, fire and herbivory is hard enough to I've forgotten what I wanted to say. Okay, in black cotton soil with moderate rainfall, fire herbivory is hard enough to influence the monodominant Acacia drepanalobium, but their combined effects do produce a dramatic decline in this tree density. So why the black cotton soils? These soils are very productive, and they do support more grass biomass. They do support more grass biomass that attract a large density of herbivores and also makes these areas more prone to fires. And the reason as to why this tree species is able to survive within these dynamics is the fact that it has a thick bark and their coppices do very well after fire. And these tree species do produce swollen thorns that host four mutualistic ant species, where the ant produces nectar to nourish the ants, and the ants act in the resistance of herbivores protecting these trees. And some previous studies from these systems have shown that fire causes a shift in ant occupancy, which makes these trees vulnerable to herbivore damage, especially by elephants. So the key question was to examine to what extent these drivers interact to influence growth and mortality. And this work is an insight from the Kenya long-term exclosure experiments that are found in Laikipia ecosystem in Kenya, particularly at Impala Research Center. The exclosure experiments were established in 1995, and during their interception and even for decades before, fire has been absent and were only recently introduced in 2013. So that's how the experimental design looks like, and these plots are found in a homogeneous black cotton soil that is dominated by about 97% of the woody vegetation, and they have 6 different herbivore treatments. And these herbivore treatments are replicated 3 times, and they are a combination of mega herbivores, mesoherbivores, and cattle. And within each of the herbivore treatments, we have a small subplot of 30 by 30 meters that have been burnt at different fire frequencies. So what are these interactive effects that we found? We found that within the plots that had more fuel load and those that were accessible by mega herbivores, there was more mortality rate in burn than non-burns. And we also went ahead to compare plots that had been burnt once and those that had multiple burns. And in this case, in the cattle plots, cattle seem to have maintained to ameliorate the effect of frequent fires by maintaining the herbaceous vegetation, which reduced the fire intensity. Within the first 2 years after burn, there was a significant reduction in tree height, and even after, which was followed by a gradual increase in tree height, and even after 5-year survey periods, these trees had not regained their original tree height. We also compared the growth rate of coppices within single and repeated burns, and in this case, mega herbivores did suppress the growth of saplings, and they did mask the effect of fire frequency. So the takeaway point is that both the mega herbivores and fire presence increase tree mortality, and interactively they do produce feedbacks that are not simply additive, and trees that survived previously burned subplot were less vulnerable to subsequent fires, and the fact that coppices in previously burned subplots regrew faster than those that had not been exposed to fire previously. A big thanks to the collaborators and people I worked with, those who made this work a success, and thank you for listening.
Thank you, Mary. Our next speaker will be Sandra, who will be telling us about fluctuations in driver dominance.
Good morning, everyone. Over the last few days, I've heard many of you touch on the spatial and temporal dimensions that add complexity to understanding in ecological systems. And I'd like to share with you today some of the work we've done on trying to tease apart those complexities in the context of environmental drivers and how their effects are likewise spatially and temporally dynamic. Now, I'm going to be very brave and give away my age here, but I've been in the park living and working for more than 2 decades, so don't laugh, but here's the proof. Some of you might recognize Andre Potgieter there. I'm just going to go off that slide quickly. But one of the constants that I've noticed over time is change. Change is part of the system here, and it's a natural part of the system. And understanding the nature and the drivers of that change is what really interests me. And although I'm still working and living in Kruger, I'm now part of the mathematical biosciences team at Stellenbosch University. You might have heard from some of our students already, where the bio stands for biodiversity. And we're a unique group of people that are from a diverse backgrounds, including ecologists, mathematicians, some engineers, and physicists. And we're all working together to look at how we can unravel this complexity within ecosystems. And we bring to the table a diverse range of skills and knowledge. So to give a brief background, which maybe isn't necessary for this audience, but we all know that ecosystems are inherently complex. And they're characterized by dynamic interactions between multiple variables, but also across multiple scales. And this complexity extends to the drivers of those systems, in that they themselves are equally dynamic. And I think we often forget that when we're starting to study a system. And keeping this in mind when we're modeling ecological responses, it's important to keep in mind that those responders, their behavior will change from one point to another and from one moment in time to another. And that's why I think it's really important to incorporate the spatial temporal variability into our models to accurately capture the fluid nature of ecosystem dynamics. So for example, I work a lot at a macro scale. So if we're wanting to look at landscape pattern and we make that one of the ecological responders that we're looking at, we'll have to take a lot of different environmental variables that we think can explain the maintenance and the formation of these patterns that we see. Things like rainfall, temperature, vegetation, herbivory, herbivore distribution, and densities. But we need to remember that it's not just the spatial location at which we record these different variables that's important. It's also the different scales at which those measurements were taken that we need to keep in context when we try to understand the results that we get from that. And another dimension is added furthermore with the temporal dimension. And we often forget that that too has a scale to it. You could have a monthly, a seasonal, a yearly. There's different scales to time as well as space. So I put this in for Chris. Numerically, we can easily represent this as a multidimensional data frame or a matrix where we have our environmental variables that are at a specific position in space at a specific scale. As soon as we add in the temporal dimension, we get a data cube where each of the different cells represents a specific location and a specific scale, and then the other dimensions can be the different environmental variables that we've got, but then also the different time frames that we've recorded those measurables at. The tricky part comes in when we start to have to understand the dynamics of these variables. So all of them are operating at these different scales in time and space, but the interactions between these variables are also complex when you start to try and tease them apart. So if we start by trying to understand landscape pattern in Kruger. And we've got it down that we need to keep in mind the spatial and temporal components of it. We then need to go and break the system down into different environmental responders and explanatory variables. And we can go through an iterative process where we think we understand exactly what's creating these patterns in the system that we see. So we take all of these deterministic variables and we bring them together in a way that can help us explain the different patterns that we're seeing, the variability within those environmental drivers and the variability within their interactions are creating the variability that we see within our system. And if we start at the bottom and I try to build this picture for you, we can begin with abarctic realm, where things haven't changed that much over time. Things like topography, geology, soils. This is within, this is not longer term time frames. And some work that we did on this, not surprisingly, we found a lot of complexity that we missed when we tried to analyze these systems in this way. And we've created an index that we call the complexity index that actually took the accumulative difference of the inverse model fit for the data that we put in, looking at change over time with Landsat imagery. And if I can get the laser, you can see here in this map, the green represents areas that are highly complex. So we didn't capture a lot of the models did not capture a lot of the variability using the explanatory variables that we had. The red are areas with less variability, and the yellow are the sweet spot. That's where we could actually find we could explain the most variability using the environmental variables that we used. But clearly, we were missing a lot of the dynamic nature of the system. So the most obvious thing for us to do was to add in rainfall. So that's one of the dynamic systems, the dynamic components in Kruger, that's a major driver within savanna ecosystems. And not surprisingly, we found that it is both spatially and temporally dynamic. And there's been a lot of work that's been done on looking at how temporally rainfall has changed. And it's been quite difficult to actually pull out trends when you look at it from a purely temporal perspective, because Kruger is such a large area, you're essentially taking all those stations, summarizing them, and looking if you can find a change. And I put this in for Corley. So this is a good example of where there are changes happening, but you don't see them unless you look at them both spatially and temporally. So there's a lot going on this page here. I just want to draw your attention to these last two plots there. They might be a little bit unclear, but they're essentially, these two as well. It's showing you on this side, south to north, and this west to east, and on this axis is month, and this axis is year. So it's essentially showing you the change over time with the trend, this top, and with the seasonal variability. So we can see in the south, there's a lot more rainfall happening in the wetter season than there used to be in the past. The central regions, there's a lot less. If we go from west to east, similarly, the eastern areas are getting more rainfall in the drier season than they were in the past. So there are subtle changes happening, but if we don't look at them at the right spatial and temporal scales, we miss these things. And not to mention, if you start to think about the impacts that each of these variables have on the environment, this will have a knock-on effect on things like surface water, and obviously vegetation phenology, and structure and composition. And we looked at that, and we looked at productivity and structure, and I'm starting to sound like a very repetitive story here, but it's variable. It's variables over space and time. And if you start adding all these things together, you start to realize, what are the knock-on effects of this? Because I've just told you that we're having these interactions between the variables. It must have a knock-on effect throughout the system. And specifically, vegetation phenology, water is going to have an impact on herbivore species. So we looked at the spatial temporal distribution of elephants and looked at how they changed. Not surprisingly, they move all over the place, but not just from a density and distribution point of view, also from a herd structure point of view. So this example here, the red colors represent herd-dominated areas and the blue represent bull-dominated areas. So you can see that in itself is changing. It's moving all the time. And that's not necessarily an unnatural process, but it is a process that we need to understand in the context of all the other environmental variables that we have interacting within the park. We could take that a step further and look at the accumulated differences, and here we can see the darker blue areas are areas that are becoming more bull-dominated, and the darker red to black areas are areas that are becoming more herd-dominated. So there are changes happening. The last thing we need to add in here, I think, is disturbance. So fire is one of the larger disturbances that are happening in Kruger, and without adding that in, we're missing a big part of the story. And once again, it's spatially and temporally dynamic. And you might notice going through this animation, there's a few years where the fire looks abnormally blocky. This is where management comes in. Management also alters the systems that are at play in Kruger. During a period where they had a rotational burning system, they would burn blocks of the park. Later on, they had a more laissez-faire approach where they would drop a match and allow the area to burn naturally. All of those things come together. So how do we bring all of these together? In this study, we used piecewise structural equation modeling to bring in the spatial and temporal components of each of these different environmental variables that I've taken you through. And we looked at how those influence landscape pattern, landscape variability. We broke it down into temporal variability as well as spatial variability and the combination of both. The different variables that we added in, I've gone through each of them, but it's just a quick show of them. We also had distance from river as one. Now, there's a lot of results on this page. I don't expect you to read all of it. I'm just going to pull out a few. The important point to note here is these are the response variables, and these are the explanatory variables. And you'll see in this block here, some switch. They can be a responder and an explanatory variable. So you can imagine a situation where a herbivore would respond to a vegetation community, but at the same time it's altering that vegetation community as it's using the landscape. And not surprisingly, rainfall from the previous year came up as a very strong driver of landscape variability. Also elephants. And I'm going to go through a few of them in detail now, but also remember that everything that's in blue is a positive effect, everything in red is a negative effect, and the darker the color, the more significant the result. So in this context, fire is associated with more rainfall. So the more rainfall you get in the previous year, the more fire you'll have. That makes sense. So when that happened, I thought, whew, at least something's working. That makes a lot of sense. But at the same time, elephants, the more elephants, the less fire frequency. Then buffalo. Buffalo densities appeared to decrease in years with higher rainfall. I thought, sure, that doesn't sound right. But then when you start thinking about it, it's more to do with the behavior of those animals, the herd sizes, the structure, because resources are more dispersed. They can now have smaller herd sizes. So that's then reflected in the results that we see. The biggest and most dominant driver of elephants was time, as the numbers have exponentially increased since about 2000. But there are also less elephants further from rivers. Then the wet season and dry season temporal variability. There's rainfall and landform are important drivers of those, and the wet and dry season spatial variability, rainfall and geographic location are important when it comes to that. Very interestingly, elephants appear to alter fire regimes by reducing fire frequencies more so than they actively select for areas that have lower fire frequencies. And similarly, the dry season temporal heterogeneity, it decreases as you have more elephants, but increases in wet season, seasonal, wet season spatial heterogeneity. Fire also tends to increase landscape heterogeneity. Yet, I've just spoken to you for the last 10 or so minutes, trying to convince you that the system is dynamic. So I have to then look at the dynamic nature of these drivers and these interactions as well. And not surprisingly, they are different. Over time, the dominant driver within the system in Kruger, it changes. You can't always say this is an area that's dominantly elephant, this is an area that's dominantly rainfall. It changes as time goes on, as those interactions change. If you were to have a look at this and just look at the accumulated difference, you can see that the areas in red are higher diversity when it comes to dominant drivers, cascading effect. And this is just a call to collaborate. I'm out of time, so I'm going to skip through this slide. I think I've made my point in terms of keeping the spatial and temporal components within your studies. And I'd like to leave you with this slide, just showing you some of the sites to look out for. Our group is quite active, and there's a lot of stuff going on this year. One is our GitHub lab. We're going to be putting up a lot of workflows and tutorials in R and Python to get you started with this type of analysis. There's also the National Institute for Theoretical and Computational Sciences, which we're part of, and also we have a research program within that. Then we also won a spot on the Global Big Ideas competition, so we'll be at Purdue in May this year, so keep an eye on that. And then lastly, we're also part of the EU Horizon program, B Cubed, which is the biodiversity building blocks for policy. And we're doing a lot of stuff there in terms of data cubes and looking at how biodiversity globally can be monitored using data cubes. So thank you.
Thank you, Sandra. Our next speaker will be Fezile, and she'll be telling us about contrasting agents of top kill in African savannas.
Good morning. I will be running through my talk on continental scale top kill regimes and tree resprouting responses. So it is quite evident from the talks at this conference, particularly in this session, that the effects of fire as a top kill agent in tropical ecosystems is well understood and to some extent herbivory. That is potentially due to its effects as a selective agent in the early regenerative stages of trees. However, top kill is widely experienced by large trees as well, and sometimes disproportionately so. In addition to fire, cyclones, mega herbivores, frost, droughts, and human harvesting are also important agents of selection in this ecosystem, and their effects have not been properly assessed. In this study, we synthesized the spatiotemporal information on top kill agents and ecological information on demographic stages that are most affected by these top kill agents. We do this in order to set up resprouting expectations in tropical savanna ecosystems or in tropical ecosystems in Africa, and to explore past and current scenarios of top kill regimes that are dominated by elephant impacts and human impacts, respectively. Using hierarchical clustering and some PCA analysis, we found that trees evolved with high fire return intervals of averaging less than 3 years and high elephant impacts in about 70% of the continent. And in addition, this can also be split into high and low drought regions. We also found another region which is characterized by droughts, but also has frost as a also occurring top kill agent. And then you can see the regions in Madagascar and coastal Mozambique moving inland. They are mostly affected by cyclones, and frost is a common top kill agent in southern Africa. But as you can tell from the previous speaker's talk, these top kill agents seldom occur alone, or they are always interacting and shifting in time. Just taking the low and high drought regions dominated by high fire return and high elephant densities, for example, we know that fire top kills small size class trees of less than 5 centimeters, whereas elephants will prefer medium to large size class trees of selected species. So in high rainfall areas where both fire and elephants are common, but drought impacts are rare, we would expect all tree species to have, thank you. We would expect all tree species to have high resprouting abilities in the early stages of their life in order to escape the fire trap, but then only those that are preferred by elephants would retain this ability as they became adults. However, in areas that are more prone to droughts, oh sorry, I forgot this moves. In areas that are more affected by droughts, the last image, we expect all trees to maintain their resprouting abilities throughout their life history. So this is just an example of the resprouting abilities we would expect across the different top kill regions. We further examined which pixels had a higher than average exposure to more than one top kill agent. We looked at top kill exposure in the past when human impacts were low, and elephants were dominant and unconstrained. We compared this with the current scenario where human impacts are dominant and elephants have been extirpated in most areas and constrained in protected areas. We found that approximately 18% of the region is now exposed to one additional agent due to addition of human impacts in the current scenario. So that is, these areas are currently exposed to both high human impacts and high elephant impacts. And then approximately 21% in the blue, dark blue, of the region is currently exposed to one less top kill agent, meaning that these areas were previously exposed to high elephant impacts, but have lost elephant distributions and do not have more than average human impacts. In summary, our results help to explain the global nature of resprouting adaptations, which could be evidence of past disturbance regimes and help guide insights into how best to rewild and manage both tropical and non-tropical ecosystem. And finally, we can see that humans have the potential to replace important ecological functions in areas where elephants have been extirpated. But to fully evaluate this, it would be necessary to have better insights into levels of tree harvesting that are ecologically representative of past elephant damage. Such information also opens up avenues for discussing ecologically sustainable tree harvesting practices. I would just like to thank some of my data contributors and the organizers of the conference for removing our notes. Thank you very much.
Thank you, Fazile. Up next, we have Jakub that will be telling us about how fire facilitates ground layer plant diversity.
Hello, I'm Jacob. I'm from the University of Edinburgh and the Royal Botanic Garden in Edinburgh. I'm really excited to be here today. I will probably have to re-watch this session because there were so many amazing talks that I want to more focus on, but also to listen to what I actually said during this presentation. So today I would like to present our research about the effect on fire on the ground layer plant diversity in the Zambian Miombo, which I conducted along a great group of colleagues from across Zambia, South Africa, and the UK. So the leading research question that I would like to focus on today is looking at the impact of different fire regimes on the ground layer plants in terms of species composition as well as species richness. I will also look at how these impacts look like from the perspective of different functional groups which are present in the ground layer. So the ground layer plant diversity is an important component of savanna ecosystems, making important contribution to ecosystem services. We've heard a lot about like how different species are important for the herbivores, but also to people culturally, etc. However, ground layer is highly understudied. More often than not, the analyses of savanna plant community composition and richness are focusing on woody plants or grasses. Consequently, analyses prioritizing one component of the flora will likely lead to potential misinterpretation of the ecosystem dynamics, where different plant taxa and functional groups which comprise these ecosystems may have different responses to environmental changes, such as, for example, changing fire regimes. So the ground layer, plant diversity, I was reading the previous slide. Fire shapes many savanna ecosystems worldwide, such as we see in the Kruger National Park, where it can promote landscape heterogeneity and maintain open grassy environments and open canopy, in a very simplified explanation. And a lot of work about the impact of fire on the ground layer diversity has been done across South Africa, Cerrado, Australia, or North America. However, across the Miombo, a region which covers up to 3 million square kilometers, depending on estimations in Africa, we are missing detailed studies on the ground layer and relationship with fire. So the Miombo ecoregion is an epicenter of land use transformation and an expanding agricultural frontier. And there's still much to understand about the ecology of the region, which is urgently needed to inform evidence-based land policy and management. So there are currently developments of carbon offsetting and sequestration schemes involving fire exclusion or policies planting trees. But what will happen to the ground layer diversity when these other aspects, such as carbon storage or tree cover or tree diversity, are prioritized? And consequently, fire is excluded, or maybe like one specific fire seasonality is being applied. So here we are addressing this knowledge gap, and we decided to quantify how the ground layer diversity of plants will change in response to fire in a fire manipulation experiment set up at Mwekera in Zambia. So we work here in Mwekera at a site where a fire experiment has now been set up for 64 years. So the experiment consists of 3 sites of 0.4 hectare each, with each one representing one fire treatment. So 2 sites are burned annually, so that's the early fire during the early fire dry season, and then the late fire in the late dry season. And also 1 is left unburned. And so the late dry season fire will be of much higher intensity because the biomass will be much drier, and the early will be much more patchy and less intense. Also, I wanted to note that I'll be using these early, late, and no fire, in the next slides when I'll be showing plots. Okay, I'm not sure what's happening. So you can see now that these plots are completely different, but more than 60 years ago, it was homogeneous vegetation. And unfortunately, this experiment is unreplicated, so it's only one treatment, only one site per treatment. But other similar experiments in the region, such as, for example, one in Ndola, has been abandoned altogether. So the one in Ndola has been changed to a football stadium. So this site contains a wealth of information which can be used to understand the relationship between vegetation and fire. So in this research, we used the experiment in Mwekera, and we set up 3 global grassy group sites, 1 in each fire regime. So on the left, you can see the GGG design. This layout has been used in more than 600 sites across Africa, Southeast Asia, Europe, and others. And so in each site, we have these 21 circular plots. That's the blue dots. And that's where we sampled diversity. We identified all species within each plot and repeated that 5 times, so you can see it here, 5 times throughout the wet up until the early dry season. So we also separately did a survey in 2022 of all tree species found across these sites, not within the plots, but across the entire sites. And we identify all species based on collected herbarium specimens and assign them to 7 functional groups listed on the slide. I know not everyone will agree with them. We have the C3 and C4 grasses as a quite simplification. We have the sedges, we have non-gramindoid monocots, so these would be forbs, monocot forbs. We have the dicots, also mostly forbs. We have geoxyls, so that's you might know them as geoxylic saprophytes, so that's dicots with substantive, long-lived below-ground structures, as well as ferns. And finally, using these species data, we analyze species composition using beta diversity metrics and Euler plots, and species richness in the ground layer using generalized linear mixed models. So coming to the results, first with the species composition. So we found 140 species within the ground layer, and only 16 out of them has been shared across all of the treatments. We also found that there is much more turnover than nestedness within the sites. So when you make a comparison between n of the plots or also across all of the plots together, you can see that the differences between them come from the fact that there is new species coming in and some species disappearing, instead of having these nested patterns where you would expect one of them to be richer and then the other one to be simply a subset. So you simply lose species when doing the comparison. Only at the early and no fire comparison, we can see that this nestedness and turnover is quite similar, and I'm going to come back to that later. Also, the late and no fire treatments are least similar. We have very high Sorensen dissimilarity, and the early and no fire treatment were most similar, so it's the lowest score. And to analyze species composition further, we looked at the Euler plots, which help to visualize these overlaps in the species composition across different functional groups. There's a lot of happening here, but if you look at them, all of them together, you can see that all of the groups display different patterns, from like much more turnover to more nested patterns. When we zoom in, we can see the pattern for the overall ground layer species composition. As I said on the previous slide, the late and no fire are the least similar. You can see there is not many species shared between them. And then the early and no fire are much more similar. The late fire also had the largest number of unique species, so we had 40 species, and then we had only 9 unique species at the no fire. No fire is, as I said, it's more similar to the early fire treatment, and to an extent we can see that it's largely a subset of that early fire site. A different pattern is found for the C4 grasses, so we have a very high diversity at the late fire plot and many unique species, and only 2 species at the no fire, which is also largely is fully a subset of the other sites. We can also observe a complete turnover, such as we found for sedges, with no species shared between the late and other treatments. Or a fully nested pattern, which was found for ferns, with all of the species being found at the early fire treatment. So interestingly, among trees, each treatment has some unique species, although the least are found at the no fire treatment. That's simply regarding the diversity, not the biomass. And early fire had the most species overall. Coming to the species richness, there is a pattern of increasing richness with fire intensity. So the late fire has the highest number of species per plot. So that's how many you see within this one more small circular plot. And it has twice as many species as no fire. So we have many less species found in each plot. But the pattern of species richness differs among functional groups. So, for example, for the C4 grasses and geoxyls, we have this much more pronounced effect. So we have a lot of C4 grasses at the late fire treatment. You have, on average, 3 C4 grasses and 2 geoxyl species in each plot. It's 1 meter in diameter, just for reference. And then at the no fire treatment, these groups are almost completely absent. And there was also no difference in other groups. So for the non-graminid monocots, the dicots, the ferns, there was no significant difference. And to kind of summarize what I've been talking about so far, both fire has been playing a very powerful role in managing diversity and the vegetation structure across the Miombo. We can see that they have been largely altered across these 3 fire regimes. We have this very open, very grassy late fire treatment, through this more patchy but still grassy early fire treatment, and finally to this most closed ecosystem with no grassy layer at the no fire. Composition has been highly altered with big turnover, especially between late and other treatments. only the no fire is similar to the early fire. So even though you don't use the fire at all, or you use it annually, in the early dry season, these systems were more similar to each other. Also in the no fire treatment, we did not find any evidence of refuge or colonization of fire or disturbance intolerant species. Additionally, it is important to note that in terms of the diversity, the no fire had the lowest number of tree species. So of course that doesn't translate into biomass, but you can see that in all of these plots we had some unique tree species, but not as many in the no fire. And finally, the richness is increasing with the intensity of fire, and it differs among groups. So looking at all of them together would not allow us to actually distinguish these responses, and we could see how C4 grasses and geozoals are most benefiting from fire in this ecosystem. And of course, this experiment is unreplicated, and perfectly we would now need new experiments to actually confirm this pattern and allow more robust analyses. But this is what we've got, and it's very important evidence. And what we should take away from this is that any homogenization of any of the fire regimes can likely have negative impacts on the diversity. Even without replication, these results are similar to other ecosystems worldwide, most in the sense that fire exclusion has a negative effect on both species composition and richness in fire-adapted ecosystems. What can be done though now is, with this experiment that we already have, is to answer questions related to temporal changes, looking at which species coexist with each other, or for example, which traits are associated with different fire regimes. And this is what we're actively working on, which we hope will further advance the understanding of the ground layer in Miombo. So yeah, thank you very much for listening. I am very much grateful to all of the colleagues because everyone was involved in different steps of the projects, from planning to data collection to identification and then the analyses. Also thanks to my funders. And yes, please chat to me. There's not much time, but please chat to me if you would like to know more about the fire in the Miombo, or you have like some useful insights, or if you're interested in the global grassy group protocols. Thanks so much.
Thank you. Up next, we have Lawrence that will be telling us about the future of large savanna trees.
Good morning, everybody, and thanks for the opportunity to speak here again. Like Sandra, I've been in this landscape for about 2 decades, but I'm certainly not going to share any images of myself. But I kind of missed the early days of the network meeting where Kevin Rogers would stimulate a little bit of debate, but then this morning Mike Anderson decided to discuss some of his work, and so I feel duty-bound to defend our elephants. So just a bit of context for the work that we've been doing on plant demography is what we've now termed the paradox of conserving elephants. So where elephants are not protected, they're in precipitous decline. But where they are protected, there's an emerging argument that they are starting to have an influence on biodiversity. At the heart of this, the canary in the mine would be large trees in savannas, and plenty of work has been done on the importance of large trees as keystone structures in savannas in particular, through a variety of mechanisms. They're important for bird communities, whether it's the work on vultures by Aramonogem and this idea that vultures are fundamentally dependent on spinescent trees for nesting, through to the Martin Cody-like work looking at change in vegetation structure and therefore habitat complexity, through to understanding the sort of trickle-down effects on vegetation communities. So this significant turnover in grass communities beneath the canopy of trees, or when disturbances removed, the nucleation effects around the base of trees. But in essence, trees are key to the fundamental ecologies of savannas. When this work was first presented, in fact, in the very first network meeting I attended, there were a couple of us that were a little bit skeptical of this, because there's a famous article called Sebachrome Blue, which talks about the bias of a photograph. Of course, it's fine if you've got 2,000 of them. And we were a little bit critical of Holger's work, because we thought, well, what about the demographic profiles? What's happening outside of that image, and what's happening in the understory? Well, turns out he was right, and so we had to eat some humble pie. Of course, the LiDAR emerged, and we're looking at tree loss rates of anywhere between 0.5 and 10 trees on a sort of biannual basis. And so certainly we have seen a loss of trees. So I decided to put money where the mouth was and to start looking at regeneration dynamics of many of these overstory dominants. And we've chosen to use plant demography as a framework for understanding disturbance in these systems. We feel strongly that at various stages in their life history you can experience different ecosystem determinants, and so it's worth considering all the various life stages. It, of course, leads to a reasonable understanding of species resilience and therefore ecosystem or community resilience. But I think there are two core elements that are worth considering. The one is there's what's called demographic storage, the idea that the longer lived or the bigger individuals can store their demographic potentials. So if you're long lived in the landscape, elephants, baobabs, you can outlast the bad period and reproduce thereafter. And then the other would be this world of the neighborhood size, this idea that well-dispersed plants play a different game than those who don't disperse at all. We know a lot about the adult stages and the loss of trees. There's an old song, and we've plenty of people published on elephant bark stripping and subsequent burns, and then you see these disastrous states. Being a tree lover, of course, I weep for certain individuals, and that tree is still standing 20 years later in that state. There's also some surprises, so the effects of drought. So elephants often painted as a bad guy, but of course drought effects, and more recently a better understanding of the effects of wind as well. The problem is that we don't really have a clear sense, or I don't have a clear sense of what's happening in the regeneration niche, and that's a terrible place for a replant to be. And so this work, and it's data that I've extracted from years and years and years of students on their hands and knees looking for seedlings with me. Oh, by the way, I offer a 1,000 Rand reward for any true seedling in the Kruger National Park except for Marilla, and I'll tell you why in a second. So the first is, wondering why there's so few seedlings in the landscape, and how do regeneration dynamics vary over disturbance gradients. So our key protagonists, knobthorn and marula, these are all trees that are familiar to you. And I've chosen them, or we choose to work on them because they're very long-lived, and their ecologies are interesting. So the idea is that knobthorn can live up to about 700 years, marula up to 300. Not much in the way of dispersal in the nigrescence. They produce plenty of small seeds, but they would rely on propagular pressure in the landscape, so it's just to swamp the seed predators. And you'd consider them to be fine-grained. In other words, whatever you see in the canopy, you see in the understory, and they would rely on the storage effect, whereas marula, slightly shorter-lived, are both dispersed, and their germination is enhanced by elephant feeding and, of course, subsequent burial by squirrels. And so they have enormous neighborhood sizes relative to nigrescence, fewer large fruited seeds, and they would be considered to be coarse-grained, and they rely on this large neighborhood size. So we started doing some smaller experiments, and then we scale up to the landscape. And so we were curious how, what the fates of seeds and seedlings might be across a gradient of disturbance. So we used our exclosures in various locations. So this would be the control site where all the herbivores are available inside the large mammal exclosure, and we built these somewhat bulletproof small exclosures for the rodents. And having just told you that, or you guys probably know this already, but marula's dispersal and germination enhanced by elephant feeding. We have marullas in 3 states: fruits, those eaten by, or the fruits eaten by impala or warthog, and those that pass through an elephant. And then the nigrescence, and we repeated that above and below ground, and we had 10 replicates of 10 seeds. And these are the results. There are 3 take-home messages here. The one is this enormous space over here. Virtually no germination in all of our trials. So on the x-axis, we've got this various treatment. We've got marula above ground, below ground, and then nigrescence over there. And here we have percentage germinates, and virtually nothing germinated in our trials. Key to note with marula is that germination was significantly enhanced if it passed through an elephant and if it's protected from rodents. and nigrescens. And to our surprise, we'd imagine that if nigrescens were buried, they would also germinate better. Not at all. We saw no germination beneath the ground, but the best survival and germination when protected from rodents. We engaged further in this problem with a fabulous group of students in Eswatini, and I mean, it's a hyper-complex experiment, but I think what's important here, the take-home message, is these are seeds and germinants, both seeds and germinants and nigrescens in 4 different states. You've got the control, we controlled for small mammals, we controlled for birds, and we controlled for insects. And not much happened in the wet season when we repeated our experiment, but what's key to notice here is that the key agents of seed predation would be the rodents, although there was a small effect of insects as well. But rodents seem to be a key bottleneck for many of these seeds, but also the germinants. And where that came from is we tried some experiments in the Kruger, and the squirrels kept eating our seeds, but then as soon as they germinated, they ate those as well. Okay, so that gives us some sense. It's not the whole story of the germination realm, but at least some sense of what the demographic bottlenecks would be. And now we scale up our thinking to some of our work. We're comparing plots in the Kruger National Park with plots in Eswatini, same soils, same climatic envelope, just different drivers. In the Kruger Park, significant herbivore effects as well as repeated, frequent, relatively intense burns, and then sites in Eswatini that have had no elephants for 120 years and very sporadic fires. And we spend months of our time on our hands and knees enumerating quarter-hectare plots. And these are just some of the data. If you think about demographic profiles and this inverse J-shaped curve as being an indicator of healthy regeneration, then it looks to us like knobthorn is doing reasonably well. Marilla, plenty of regeneration individuals in the landscape, but very few adults. The key here is the change over time. So we've enumerated these plots over some 11 years, and we've seen about a 90% loss in the big trees over that period. But demographically, it looks like knobthorn should be fine. What is really fun about this work is now getting onto our hands and knees and looking at, putting up fences and manipulating these systems. It turns out the only true regeneration we saw, the only true seedlings were from the Marillas. And probably 15% of those regeneration individuals were true seedlings that had germinated that year. Another 25% of those had germinated in the last 3 or 4 years. So they were small gullivers, they'd been eaten, but they're resprouting. So despite the lack of large individuals, seeds and fruits and seeds are being imported into the area. So the lack of regeneration large adults doesn't seem to be that much of a problem. Knobthorn, on the other hand, which we know rely on propagular pressure, and they have a very small neighborhood size, we found no seedlings, hence the 1,000 Rand reward. We started to look at some of these dynamics, and as I said, we've seen a decline in the density, but we've also seen a decline in the height of these trees. So the average height of trees larger than 10 centimeters declined quite significantly over that time. And what we found, and this on the x-axis you've got height, and the y-axis is seed production, is that we find that there's some threshold height that you need to get to in order to produce seeds, flowers and seeds. Some bright sparks said, what about the giraffes? Maybe they're eating the fruit. So we said, fine. We measured all the trees on the inside of exclosures, and a relatively similar pattern emerged. So there's a minimum height requirement to producing flowers and seeds in nigrescens. We skate over to Eswatini, no elephants for 120 years, and very few intermittent fires. And again, you get this wonderful inverse J-shaped curve. But Marilla was a very sad story indeed. So nigrescens, very, very high densities of the large individuals, so plenty of seeds in the landscape, and plenty of seeds. So this is the only part of the world where I've found true nigrescens seedlings. And so it seems to be in a healthy shape. The marula, on the other hand, very, very low densities, and that there might be some legacy effect there. Maybe that area never had marulas in the first place. But our argument is that due to the lack of elephants and also squirrels for some bizarre, I still don't know if somebody can explain why there are no squirrels in Eswatini, feel free to do so. But in essence, very few large individuals and no young seedlings. Just a quick sidebar is we are starting to see the effects of bush encroachment now in impacting the seedlings. And so, from the small-scale germination experiments to thinking about landscapes, the following pattern seems to emerge. Basically, it's a really rough time to be, or it's a rough thing to be a woody plant in the savanna, get absolutely pounded. Knob thorns in the Kruger, we're somewhat concerned, certainly in those basaltic soils in Lower Sabi. But the fun part of that is that if you, just hearing Sandra's talk today, just thinking about heterogeneity in the landscape and heterogeneity of drivers, whilst we might have found these sterile landscapes in Lower Sabi, lo and behold, later that year rolled into some of our plots on the granites and plenty of taller trees, and they're all flowering and fruiting much shorter. So the idea is that these patterns are heterogeneous in the landscape. As far as Eswatini marillas go, I think in the absence of some of those mutualists, we see a fundamentally different picture. And so, damned if you do or damned if you don't. But in essence, disturbance regimes do matter. So it's accumulation of, as I say, 10 years of plenty of students on their hands and knees looking for seedlings with me, and I'm deadly serious about that reward for a true seedling in the Kruger Park. Thank you very much.
Thank you, Lawrence. Next up, we have Joris, who will be telling us about elephant impacts and above-ground carbon stocks.
Thank you. Can you hear me well? Yeah? Cool. So as Lawrence, I need to refer back to this morning and to Mike's talk. I'm going to talk about elephant impact as well. I'm not sure if I can show you that our elephants are stronger than his, but I think I will show you that our trees are stronger than Mike's. So that's kind of the main conclusion of my talk. Our trees are stronger than Mike's trees. And I will show you why I'm saying this. And we've been relating this actually to carbon stocks as well. So I first want to highlight Sipe Sigle and Bongwa and Camille Schrucker. They are two students that the work that I'm presenting is their work, basically. They're both master students. Sipe Sigle was a master student at Nelson Mandela University, and Camille was a student at SLU, my university in Sweden. I just want to go back to a bit of a bigger picture to set my talk. Last year we published a study. I was only one of many. This work was really led by Oswald Smits from Yale University. And we discussed this opportunity of restoring wild animals as a negative emission opportunity. So negative emissions are basically about sucking carbon out of the atmosphere or out of the air. And there's many people that suggest that we need to do that in addition, obviously, to reducing our emissions. And one of the main ways one of the main tools for negative emission at the moment that's being used is planting trees. I think many of you are aware of that. So tree planting campaigns are a negative emission solution. But we looked at this alternative opportunity, and that's restoring animal populations. And basically what Oswald Smits and his lab did is calculating for these 9 different animal taxa, how much carbon do they add to the system if we would restore their populations. And we basically concluded that just for these 9 taxa, we would be able to reach 60% of the current annual emission target if we would restore their populations. You can see some of them. There is an elephant there, but that's actually the African forest elephant, so that species was included. The one that was not included here was the Savanna elephant, and Savanna elephants arguably can have a big impact on above ground carbon stocks, and we've seen Mike's example. His very strong elephants had a massive impact on trees. So we were interested in looking into that, the impacts of elephants on above ground carbon in an ecosystem. This has been done before in this park as well. Andrew, sitting in this room as well, has looked at this in Kruger National Park, and they showed a pretty strong reduction of carbon by especially elephant bulls in the regions that they visit frequently. There's also been some work in northern Namibia by Sandhage Hofman, and I believe Duncan was in the room here also. He was involved in this study. They showed the same thing. So you basically see here that with an increasing elephant density, you see a reduction in above ground carbon, woody carbon. But what they actually showed is that this is being compensated by an increase in soil carbon. And they explained that basically by the dead wood that's resulting from elephant impact is being taken up in the soil and stored in the soil. So they basically conclude that with increasing elephant density, the net effect of elephant impact is kind of neutral, possibly even slightly positive. And there's some more work actually that show that for Kenya and Savanna, they actually show the positive impact of elephants on the ecosystem carbon. So we were interested in studying the same type of question in a different park. And first of all, we were interested in looking at this kind of quite diverse ways of how elephants can potentially structure a savanna. So this is kind of the pathway that Mike has been showing you. So they basically reduce the number of trees in the landscape and can convert the savanna in a much more open savanna system. But there's this alternative way that has also been suggested, and that is that actually, yes, they do remove the tall trees, but a result of that is that you see an increase in smaller trees and shrubs. And this actually, both these pathways were already suggested by Richard Bell I think in the late 60s or early 70s. So that goes back quite a long way. So what is the net effect for above ground carbon for these two pathways? That's one of the questions we had. And then secondly, you can see here, in this state, you see a lot more multi-stemmed woody individuals. So not the single stem one as you see here, but a lot of multi-stemmed woody individuals. And what is the effect of this increase in multi-stemmed individuals on above ground carbon estimates? And these are actually quite often missed in ecosystem carbon estimates. Secondly, we were interested in the differences in species. So elephants can also induce a shift in species composition, so they can change the species composition. And as we know, tree species can vary a lot in terms of the density of their wood. So you have softwood species versus hardwood species. So we were interested in specifically testing for if elephants change the species composition, does this then actually also through changes in wood density maybe affect above ground carbon? So yeah, we were basically interested in these two questions, as I said, and we were testing this in Gekloe in Flosie Park. I will refer to this as HIP in the rest of my talk. HIP is in the northeastern part of KZN, and elephants went extinct there during the late 1800s. They were reintroduced from the early 1980s, and since that time they've strongly increased in numbers. So during our study period from 1999 to 2017, they basically increased from about 250 to 750 elephants, or perhaps close to 1,000. At the end there were about 1 elephant per square kilometer in HIP at the end of our study period. So what we also have in HIP is this long-term monitoring of elephant impact, and not just elephant impact, but also woody plant monitoring. So this was started as part of a big effort by Dave Balfour in the late 90s. And then later on we took this over as a research project. But I'll be talking about the data from 1999 to 2017 here, so it's basically close to a 20-year period. And you can see here the distribution of the plots in the park. So they are well distributed across the different wooded systems of HIP. So basically, one plot is 50 by 50 meters, and within that plot, people record every woody individual, although it's a nested design, so the smallest seedlings are only recorded in this, what did I say there? Well, you can basically see the different size classes here. So there's 4 different plot sizes, from 0 to half a meter. They are measured in the small plot size, and the higher than 4 meters they are measured in the full plot. And then you see these 2 intermediate size classes are measured in kind of this plot size here and this plot size here. So hopefully that makes sense. Sorry, that's a very poor explanation. And then for each of the individuals, so for these different height classes, the species is being recorded, and next to the species, the number of stems, and then for each stem, the height and the diameter. But note here that the diameter is measured in different height diameter classes. So these different diameter classes, 6 in total, and for each stem, these were the measurements. And in addition to that, for every woody individual, the elephant impact was recorded according to 3 different classes. So as the percentage of trees toppled in a plot, the percentage of branches broken, and the percentage of bark stripped. So to start showing you some results, so these are results from Cypress Ziegler's thesis. So first of all, we can see here that over this 20-year period, we see a very strong reduction in tall trees. So it's maybe not that obvious here from this box plot, but basically the number of tall trees in a plot on average is halved. So from about 200 or close to 200 trees per plot to half of that in this 18-year period, and that's actually a continuing trend. So there's a very strong decline of tall trees in the system. At the same time, that's being compensated, or we see a very strong increase in the small height classes. So very strong increase in trees from between 0.5 meter and 2 meters. In fact, also in this height class, although less strongly, 2 to 4 meters. And because of that, the overall tree density has actually been strongly increasing in HRP. So a strong reduction in tall trees, but a strong increase in smaller trees, and the net effect is a strong increase in tree density. Then we linked that to, that's obviously still not necessarily linked to elephants. So then we looked at for each individual species, at the change in numbers for that species, and we related that to elephant preference. And the elephant preference we calculated based on the elephant impact on these different individuals. And what you can see here is that species that are preferred by elephant, so they're on the right-hand side, are mostly declining actually over these years, and these are typically shrub species. Sorry, tall tree species. So a couple of your acacias. The species that are actually increasing during this period and that are disliked by elephants are mostly shrub species like Dicrostachys. Well, you see the other species there. So there seems to be this clear link between elephant preference and the trend of the individual species in terms of whether they're increasing or declining. So I think this is some evidence for elephants driving some of these processes where the system in HRP is changing from a system with tall trees to a much more shrubby state. So then we converted that into an estimate of carbon stocks, and I have to speed up a little bit, but basically what we did is we determined plant volume based on a very simple, arguably very simple anomatic relation, and we did that for each single stem. So, and then we converted that volume into biomass using wood density data, and we did this in 2 different ways. So we used wood density data for each species, so we actually measured that wood density data ourselves in the park. And then the other way was actually using just one average wood density data. So one wood density as an average for all these species together, okay? So, well, and then based on that, combining wood density and volume, we created a carbon stock estimate using a carbon conversion factor of 0.47. So that's a published carbon conversion factor. And then we linked those carbon stocks to the average elephant impact of a plot. So what did we find? So first of all, I think one striking take-home message is that actually over the 20 years, there's very little change in total carbon in the system. So that increase in tree density has not really led to an increase in carbon, and that's likely because tall trees were being replaced by smaller shrubs. That's at least how we interpret this. Secondly, there's no clear impact of elephants. So the change in carbon over these 20 years is not really related to elephant impact. So the change in carbon is very similar at extreme elephant impact compared with low elephant impact. Although this here, there's some indication for a significant reduction in carbon at high impact. But note the values that previous studies were showing. So Andrew and the data from Namibia, they were showing a much stronger reduction of carbon under influence of elephants. Okay, then we included wood density. So this is now, this is based on one average wood density value across all species. Then we included the variation in wood density. So we gave each species its own wood density value. And you can see here we had a wide range in wood density, so from basically below 0.4 to close to 1. So interestingly, including that wood density did not really change our figure here. So now you can see this is the value with just, the light green is the value with species-specific wood density. The dark green is just one average wood density value. And you can see there's no real difference between these two situations. So including species-specific wood density information has not really changed this picture. So conclusions. HAP's vegetation is changing very strongly from a tall tree savanna to a more shrubby vegetation state. So our trees are much stronger than Mike's trees, of course, in this case. Although on a side note, these are not the trees, because the tall trees seem to be losing out, in fact, and they're being replaced by shrub species. So I should say our shrubs are maybe stronger than Mike's shrubs. This structural change has meant that there's been limited change in above ground carbon, actually, interestingly, and we did not find any clear impacts of elephants on that either. And then we did see indications for changes in relation to elephant preference. So we have some evidence that elephants are behind these changes. Well, sorry, I already said this. So we don't see strong evidence for elephant-driven changes in above ground carbon. And then just to finish this, we did not include soil carbon. So if we take the message from the published papers that elephants are actually increasing soil carbon, the net effect in HAP might actually be an increase of soil carbon under elephant impact. And secondly, elephants are obviously about much more than just carbon, so we should be very careful with linking elephant conservation to carbon and carbon as a climate mitigation tool. I see my time is out. I should have acknowledged my student. This is from 20 years ago when I did my PhD in HAP, and this is me, this is my car, and this is actually a picture that my student Hilko Jansma made for the insurance report. So, thank you.
Thank you. We will now be moving on to the animal session. Unfortunately, Clinton could not be here, so we will be moving up to the next talk by Ryan, and he will be telling us about connecting protected areas to stabilize elephant populations across the meta population.
Hello, hello. Hello, everyone. My name is Ryan Wong, and I'm a postdoc at the University of Pretoria. And today I will be talking to you about the value of connectivity when we think about elephant population growth. So, since moving to Africa, I've had a chance to talk about my work with a lot of different lay people, tour guides, fellow conservationists. And I get a surprisingly common response in that everyone's saying there's just too many elephants. And this is a common narrative in the media as well. And I find this all a bit counterintuitive, given that elephants are still listed as threatened by the IUCN, and their populations were at their lowest numbers just a couple decades ago. So this kind of prompted us to ask the question, well, how fast are these elephant populations growing? How are these populations varying across southern Africa? And what are the patterns and drivers of this growth? And what lessons can we learn from these different protected areas as we think about developing these kind of long-term solutions for stability of elephant population numbers? But when I say stability, we're kind of looking for that 0% growth, so we kind of have this stable population sizes. To answer this question, we gathered 713 different survey estimates of elephant population numbers from 103 different protected areas across southern Africa between the years of 1995 and 2020. Now, pretty much all of these data were publicly available. We grabbed most of them from the African Elephant Database, and then the remaining from whatever gray literature we could find. Once we had those estimates, we calculated elephant population growth using the diffusion approximation method. Now, I want to explore these patterns of growth with you through the tale of three different parks, Addo, Hwange, and Kruger National Parks. all of which are national parks that contain elephant populations growing at about between 3.5% to 4%. And each one of these parks has garnered their own concerns about having too many elephants. But as we'll soon see, each have very different circumstances going on. So let's begin with Addo National Park. So for those of you unfamiliar, Addo Elephant National Park is a small park located in the Eastern Cape of South Africa. It's home to about 650 elephants, but the vast majority of them really only just use 300 square kilometers of the park. Addo has some very strong fences that keep the elephants in, and it's fairly far removed from any other protected areas with elephants, thus making this population effectively isolated from any greater metapopulation. Now, perhaps unsurprisingly, this really high density of elephants leads to what may be a familiar sight to many people, these really intensely used water holes with lacking any significantly large vegetation in the immediate area. And when we look at the population growth for Addo, we see that it's been growing at a relatively consistent 3.4% over the last 25 years. And it's important to note that this 3.4% is less than the maximum possible growth that can be achieved through births alone, which is about 5.5%. Now, the reason that this growth has been lower than that 5.5% threshold is because there's been a lot of active management and translocation of individuals elsewhere, thus suppressing the overall growth. These interventions are also why we see some deviations in the population numbers from kind of the average annual growth rate, but for the most part, this is relatively consistent growth. So for Addo, this is about pretty much where the story ends. As an isolated park, these elephants really can't do anything but live, breed, and make more elephants. So in order to kind of manage these numbers, the park has to kind of have these active interventions, particularly through the translocations as well as contraceptives, both of which are very costly endeavors. And when we looked at all the other isolated parks in our data set, we actually found this to be a very consistent pattern, that there's generally relatively rapid growth with very little variation. So all of these parks may ultimately end up with that, the same fate of just perhaps too many elephants unless we have this active interventions. But let's move on to our next case study, Hwange National Park. So Hwange is a very large, unfenced park in northwestern Zimbabwe, and it's connected to the kind of larger, greater Kaza region, and its protected areas or so. Now, Hwange has one of the largest populations of elephants in Africa, home to approximately 45,000 elephants. So naturally, and perhaps unsurprisingly, there are quite a few water holes that also are, very intensely used, particularly now, given an ongoing drought where all the elephants are really crowding around what little water is available. And then when we look at the population growth for Hwange, it's been growing at on average about 3.6%. But as you might notice, there are two very different patterns of growth going on here. Now, so from 1995 to 2001, the population grew at a whopping 10% annually. This is, way higher than what can be, achieved through demographics alone, through just births. What this means is that Hwange had to be receiving a large number of immigrants from its neighboring protected areas. Now, what's interesting is that these neighboring protected areas actually differ from Hwange in that they allow certain amounts of sustainable human activities such as trophy hunting, timber extraction, and some agriculture. As a result, the IUCN categorizes these as kind of 5 or 6 level parks or so, and as opposed to Hwange, which is a level 2 park. And so what must be happening during this time period is that these elephants were moving out from these neighboring buffer areas with increased human disturbance into this kind of safer refuge of Hwange. But from 2001 onwards, there's been overall relatively neutral growth. But what we are seeing is, again, these large decreases and increases in elephant population numbers, which, again, could really only be occurring from movement, but in this case, it's movement back and forth in and out of Hwange. So we want to take a look at what this, these patterns of this interannual variation, this year-to-year change in growth. So when we compare Hwange with its neighboring kind of buffer areas, we see that actually Hwange is showing a lot less overall interannual variation than the nearby buffer areas. These buffer areas, right, like Moktetsi Complex and Chovy District East, are showing much larger variations from their average annual growth rates, most easily explained by movement. And then when we kind of zoom out to the entire aggregate of all of these connected parks, the whole Casa meta population, we actually see this pattern still playing out, where the more central, well-protected areas showing a lot less variation in their numbers year to year than those surrounding buffer areas that contain more of these human activities. Their centrality and protection seems to make them kind of safer refuges, and so elephants can move in and out in response to whatever the local conditions suit them best. And when we look out at all of our different parks, we found this to be a statistically significant relationship. So on the one end, when we think about these isolated parks, they all show this consistent pattern, as I showed you before, kind of this relatively rapid growth with very little variation. But at the other end of the spectrum, we have these buffer areas which show a lot more volatility. They have a lot more freedom of movement being connected to other parks. They also have that increased disturbance, so elephants are going to move in and out in response to whatever is going on. And meanwhile, these core areas actually show this intermediate level of variation, which again makes sense as we think about that Huanggi example. Huanggi may be acting as that safer refuge in comparison to the more disturbed buffer areas, but because of its connectivity, it still receives immigrants and has a little bit more variation. What we believe is happening here is that this is a signal of some sort of kind of source-sink dynamic that's driving variability in the numbers in buffers and relative stability in the core areas. But beyond just the interannual variation numbers, when we actually think about the overall aggregate weighted growth rate for the entire Casa meta population, it's actually only 2.5%. So this leads us to our next conclusion, that we really need to think about scale when we're talking about elephant population growth, that the Casa meta population growth is different than had we just looked at Huanggi alone. And not only is it different, it is actually this wider view is showing a growth rate closer to that 0% kind of stability that we'd like to achieve. These larger meta populations just aren't seeing that same runaway growth as we do in those isolated populations. And when we zoom out even further and looking at the kind of all of Southern Africa as a whole, we calculated a weighted growth rate of just 0.16%, that there are quite a few number of sinks offsetting these sources and the growth rates there. But let's move on to our final example of Kruger and see what's kind of happening here. Now, and many people are concerned about the number of elephants that are here in Kruger, particularly when you see these really kind of barren, treeless water holes. But when we look at the population growth rate, yes, they've been growing very quickly at about 4.1%. But interestingly, we're really not seeing much variation, which is a bit odd because Kruger is connected to nearby reserves as well as Limpopo National Park in Mozambique. But so when we look again at this interannual variation in numbers, we do see that similar pattern between Kruger and surrounding areas, with Kruger showing less year-to-year variation than those nearby areas. This does seem to imply that there is some movement of individuals between Kruger and its other neighbors, but perhaps it's not at enough of a level to kind of suppress that overall growth rate or introduce a lot of variation. Now, we think this may have something to do with the park's history. The fences around Kruger only came down in the mid to late 1990s, so perhaps elephants have not had as much time to kind of find safe migratory routes or explore the nearby areas as they do in like Huanggi, which has been unfenced since its creation in the 1920s. But recent evidence suggests this may be changing. Recent surveys from nearby Ghana-Rajot suggest that elephants are actually moving out in relatively large numbers, potentially moving up into northern Kruger. And this may imply that we're starting to enter and see this new era of connectivity in the greater landscape, which provides us hope that maybe we'll start seeing this kind of lower suppression and evening out of the overall metapopulation. And this is particularly good news when we think about kind of these greater Limpopo transfrontier efforts to connect Kruger to other parks like Ghana-Rajot, Benin, and Zanav. That as we increase connectivity and allow for the movement of species, that these elephants may find more opportunities to naturally disperse in response to whatever those local conditions are. And so looking forward, we propose a number of kind of large-scale corridors that can connect our existing elephant metapopulations. So We developed this map using millions of telemetry data points from elephants all over the continent to develop maps of kind of habitat suitability that also minimizes the overlap with existing human presence and activities. Our hope here is that by working with local communities in these areas, we can design corridors and linkages between these areas to allow elephants to naturally disperse and naturally equalize their populations without those management interventions. So just to review, we saw what happened in Addo, this isolated park that has very quick, consistent growth but requires this active, costly management. Then we saw in Huanggi somewhat more variable growth rate driven by movement, but this growth is super dependent on scale, both temporally and spatially. In some periods of the time, they grow really quickly, and other times it's neutral. And that when we zoom out to the meta population, not only is the growth different, but it shows a growth rate closer to 0%. And then finally, Kruger seems to be showing signs of being in this intermediate stage of stabilizing, and that there is some interchange of movements of individuals, but it's only just starting to happen across the greater Limpopo landscape. So with that, I want to thank the team at CERU and my co-authors who helped support all this work, particularly the late Professor Rudy Van Aarde, who was also the photographer for all the lovely photos you saw here today. But with that, I'll take any questions.
Thank you. Up next, we will have Adrian, who will be telling us about African elephant foraging behavior as a novel management tool.
Okay. Good afternoon, everyone. So this work that I'm going to be presenting once it appears on the screen is really the work of my PhD student, Pat, who was here last year, who spoke about swamp wallabies, and is carrying on with our odor work and what we've done. As they scramble to get this sorted out. So this is a collaboration between University of Victoria, University of Sydney, and then North Dakota as well. Okay, there we go. Great, there we go. So there's Pat there, who's back in Australia and either sitting around listening to this with a beer, and hopefully I don't screw this up. So, right, so the one thing is I don't need to spend a lot of time chatting with everyone here about the impacts that elephants had. You can go out in the car park and watch the respective gangs fight it out after lunch. We know that elephants go along and they push over trees, they break branches. Enough of this can lead to community changes, but in areas where humans and elephants overlap, we can actually get human-wildlife conflict, and human-elephant conflict. What I'm really interested in, what we're really interested in, is the decisions that elephants make that ultimately lead to these impacts. So it's the idea of them going along and making decisions from large to small scales. So we know that they do this. They go along, they'll choose habitats. Within those habitats, they find patches. Within those patches, they'll find specific trees. So they're not going around looking for individual trees across the landscape. Right? And also, when we take a look at those patches, we see that they prefer to choose high-quality patches to low-quality patches. This makes sense. You go along, you get greater intake rates, you get better nutritional gain. Okay? So greater bang for buck, as it were. How do they do this? Well, the one thing that we've been able to show with a lot of our research, and I'm sure I've bored a lot of you with this, is really the use of odor and keying off of the odor that's being given off by plants in these patches. Okay? So understanding this, we asked two questions. If we understand that they use odor, what aspect of odor do elephants use to make these foraging decisions? And if we understand that, can we then go along and manipulate those cues to shift elephant foraging and change their patch choices? Right? So this is the conceptual model that I literally just got from Pat a few minutes ago that's been updated. So basically how this works is an elephant moves across a landscape. You see this is plant over, it's moving, and it picks up odor cues. Right? So here is you can see the high quality food is the green. It's mixed. This is also intermediate, high quality, it depends. But it has a choice between going there or to a patch that sits here with some good quality but surrounded by low quality. What we would expect is it would come up and choose either a patch, and this could be 1 or 2 trees completely surrounded by other high-quality plants. This is a great patch. Few good trees surrounded by moderate quality. This is still a better patch to be than in an area that has 1 or 2 good trees but completely surrounded by low quality. Now, understanding that, can we then replace those actual trees with virtual neighbors, with the odor, and then turn elephants from instead of coming here to up there? Then that means that those trees that are within those patches up here, they're susceptible, but down here they can be safe. Okay, so how do we do this? Well, we went to Adventures with Elephants. We worked with 6 semi-tame elephants, 4 females, 2 males. We built one of the world's largest Y mazes. You can see it here. There's an elephant, these semi-tame elephants. Here is where we have our odor that's coming there. There's a fan, I'll show you in detail, hits this wall and comes down. The distance between that elephant and those choices is 9.5 meters, okay? What we did is we used 3 trees that they come into contact with in this area. Okay? There's Papaya capensis. This is their favorite. This is their chocolate in this area. They eat that. A moderate quality is the dombea, and one that they completely avoid is the euclea. Okay? So we've got high quality, moderate quality, and low quality plants. What we then did, and I can show you, so this is what it looks like underneath that triangle. You can see a caged off area. We've got 175 grams of Papaya capensis put there. You can just barely see the fan. I'll show it in a better picture now. Underneath here is a food tub that has a proportion of whatever's sitting there that the elephants can actually eat. Okay? So we used a focal patch, was 175 grams of Papaya capensis, and we had 4 treatments. Right? One was, well, we did exactly the same. We gave them a choice between 175 grams and 175 grams of high quality. We would expect them to choose randomly and go either way. Right? The next we have is that we took that 175 grams and surrounded it with 875 grams of high quality Papaya capensis, making a super quality patch. Right? Higher availability, same species. We did the same thing then with dombea, so we still have a moderate quality, but it's still a higher quality. Then what we did is we surrounded that with euclea, 875 grams of euclea. So they have a choice, 175 grams or one of the treatments, and we randomized where they were within the Y maze. Okay, so what do we find? Okay, so here you go. So we've analyzed this using generalized linear mixed models. This is the probability of selecting one of the treatments. Okay, here is the 50% line. If something is sitting on that line, it's showing random selection. Half the time it goes one way, half the time the other. If it is above, it shows preference for that. If it's below, it's avoiding the treatment. So what you see when we compare 175 grams of Papaya capensis against itself, there's exactly the same. Okay, so they're random choice. When we gave them a choice between that and higher quality patches, the elephants selected the higher quality patches, whether it was the high quality or the moderate quality mixed in. But look at this. As soon as you surround that high quality patch with the low quality euclea, they avoid it. Okay? So they're down here, they're moving away. Okay, cool. Step 1 done. Now let's take a look at this. All right. Understanding that now euclea turns these elephants, can we then utilize the odor of euclea to create virtual neighbors? Right? So we went along and did this, and we used what's called the rules of reliability. This is this paper by Orlando et al. And how this works, I'm going to give you Odor 101, okay? Very quick introduction to this. Okay, understand this. Anything that you smell is not made up of a single volatile organic compound. Apple and banana smell very different, but they share volatile organic compounds, okay? They have some that are different as well. So what we did is we went along and took the scent samples of a number of individuals of the same species, okay? What you want is you want those species, those compounds that are consistently found in every individual, okay? And then what we did is we went along and said, okay, understanding that, let's take a look at pairs of these in relation to each other. Do we have a situation where we then have one that's, say, double the amount of the other, and it's consistently double the amount of that in the pairs and all the individuals? This increases the precision. So this is the amount of those compounds within those pairs. This is how common they are. And what happens is if you're at low and low, this is noise, okay? This is, we're not getting anything. As you start to move up, as consistency increases, precision increases, you move into what would be called a cue, and then finally the signal. These are the compounds that say and smell like that. You don't need 135 compounds, you can do it with a lot less. Okay, so that's what we did. We used consistency. We wanted any of those compounds that were found in greater than 85% of all the individuals that were there. We then used a precise proportions between 0.08 and 1, so that's high to absolute precision. Read the paper. I'm not going to give you a lecture on Orlando at all. This boiled down to the fact that 5 compounds define the smell of euclea. 5. And they're found in 4 pairs. So you can see them here: limonene, decanol, lananol, beta-pinene, and gamma-terpenine, for any chemists in the audience. Okay, so we've got that. So now what do we do? Well, we created virtual neighbors. So the first one we created was the informative. This were those 5 compounds mixed in the correct proportions of those 4 pairs. Right? So that's the first one. The next, we flipped those proportions. So if one was double of the other, we then turned around and flipped them. And we made the one that was smaller now double. So what this shows is that we're testing the compounds that are there. Is it just their presence, or are the amounts important as well? We then went along and created uninformative. Okay, so these were 5 compounds that were found periodically within the odor samples, but were not consistently there. Okay, so we mixed these together and created that. We then went along and mixed this and made 1.5 milliliters of these individual mixtures, and we put them in one of these tiny little, like, I think they're homeopathic, you get oil, so this tiny little top here. Here you can see 175 grams of picopensis. There's the fan, there's the vial. And what we did, and I'm not going to go through all of the experiments, we were able to show that the emission rate from this tiny little bottle was equal to 875 grams of the other vegetation. All right? So we did that, we had the fan running, and we then also compared these things with real plants. So what we did is we tested the 175 grams against itself again, and then created a real low-quality patch by surrounding with 875 grams of euclea. Okay, so what do we find? And this is cool. Okay, so once again, this is the probability of selecting one of the treatments when you compare 175 with 175 of focal patch. Well, they go this way and go this way. Doesn't matter, random. Now look at this. When you use those flipped proportions, okay, so we've taken that double, we've flipped it, the elephants, once again, show random. So it shows that it isn't just the presence of those compounds, it's the amount of those compounds that are important. When we use the uninformative, once again, there's no difference. If those 95% confidence limits overlap that, no difference, okay? Now, look at this. This is so cool. If you take the real euclea, you surround that with it, they avoid it. They see it as a low-quality patch. When we then take 1.5 milliliters of those compounds and put it in front, they avoid it as well. Okay? Scent alone is turning these elephants. So what does this mean? Well, the first thing that should come out, we got plenty of time. First thing that comes out is the fact that the reproduction of odor can be an incredibly powerful tool in conservation and management. Okay? You've seen the impacts that elephants can have, and what we're seeing is that what's really important about this work is that it's natural odors. Okay? There's a paper that recently came out. They're keeping elephants away from kraals and fields by hanging cloth soaked in diesel. Well, this is a novel odor in the environment, and the one thing we know about animals, they make associations very quickly between a novel odor and something. So it's going to take one elephant to walk through that diesel wall, get to the other side and find watermelons or mealies or sorghum, and it will associate immediately diesel with whatever it found on the other side. One elephant. What this does here is we're using the natural odors that are hardwired. It smelt euclea, it's tasted euclea, and it hates euclea. And if it were to happen to go to some place and think, oh, well, I can make an association, it walks back out in the felt, and it will once again come into contact with euclea. Okay? So we're using the things that are hardwired. So what does this mean? Well, this is a first step. It's small scale. It's 9.5 meters. We're well aware of that. However, the results we're finding seem to suggest a possibility of a cost-effective and non-lethal way of turning elephants and changing their patch selection. All right? We could potentially protect specific plants. If you have endangered species in the park that you're worried about being eaten by elephants, maybe we create a low-quality patch by using odor. You don't have to fence it. You don't have to guard it. You don't have to hang diesel rags around it. We also have a situation of moving into reduce human-elephant conflict, especially with subsistence farmers. Their fields are tiny. Will we ever be able to protect a citrus orchard? No. Too big, too much odor. But you could protect a person's life in the sense of their crops for that year by using odor. And then finally, we could actually improve restoration projects. So if you have seedlings that are going, or specific trees you're planting to revegetate areas that are getting hammered by herbivores, we have the possibility of doing that as well. So this is the first step, and we're marching forward with this. And with that, I will say thank you very much, and I'm happy to take any questions you have.
Okay, thank you so much. Next we have Margaret, who will be telling us about savanna elephant movements on a fenced and drying landscape.
All right, well, good afternoon, or I guess it's afternoon now. My name is Maggie Swift, and today I'll be giving an overview of the first 6 months of my postdoctoral project, which uses computer modeling to answer policymakers' questions about wildlife movements. And today I'll be using elephants as kind of a proxy for wildlife movements in general. As a quick note before we get started, whenever you see this picture of a real elephant, I'll be talking about real GPS data. And when I'm talking about simulations, you'll see this picture of a simulated elephant. Across the globe, and especially in southern African savannas, wildlife rely on the free movement across heterogeneous landscapes to access resources, which facilitates biodiversity and population resilience to disturbance. But human infrastructure and activity often inhibit these crucial movements. In particular, the management of livestock disease has often relied on these veterinary fences separating wildlife and livestock, which can have unintended consequences for wildlife. Today, I'll specifically cover the consequences for a savanna elephant in the Kavango-Zambezi Transfrontier Conservation Area, also known as CASA, so thanks to Ryan for already introducing this region. The largest terrestrial transfrontier conservation area in Africa and the world. It was created in 2011, an international agreement between 5 southern African countries to facilitate the free movement of animals across international boundaries. However, major linear barriers still limit wildlife movement within CASA. For example, in this region, highlighted here at the border of Namibia and Botswana, animals naturally move around the region to access seasonally available water and vegetation near the Okavango Delta, but veterinary fences have restricted their movements. Here I'm going to be showing some data from real GPS data from female elephants, here as a proxy for elephants traveling in herds, because these females are traveling with their young and with other female elephants, collared in Botswana in gray and Namibia in red. As you can see, the red and gray aren't mixing very much, right? So these fences are really separating these populations in the west, and then in the east they're able to mix because there's less fencing in the way. This lack of crossings by female elephants is probably due to the fact that even if an elephant, the male elephants can push right through the fences, but there's these steel cables that remain in the way for a young elephant that can't cross over, and the mothers won't leave them. Some of the general consequences of fencing for wildlife include a disruption of natural migrations, reduced resilience to drought, deaths from entanglement, which doesn't really affect elephant, but a lot of other wildlife can get entangled in these fences and die, increased population density, and increased human-wildlife conflict. And these last two are really important for elephant. As Ryan was talking about, population growth rates usually about like 4% and even less in this region, but actually in where I'm pointing out with this green arrow, it's over 5%. This is like a box of elephants, a lot of human-elephant conflict going on here. But through some work with Cornell's AHEAD group and regional partners like SADC, non-fence-based foot and mouth disease management approaches are now possible and accepted in the region. And foot and mouth disease management has been one of the main drivers of fencing. So this is really important time for conservation, that now we don't have to rely on these fences for disease management. So targeted fence decommissioning can lead to relief of high-density population pressures, like in the area I just talked about, improving wildlife population resilience, increasing sustainable tourism options, and reducing human-elephant conflict. But if you're a manager or a policymaker, you might be hesitant to adopt a new management tool without knowing its actual effects on the landscape. So that's where my project comes in. The goal of my postdoc is to provide computational tools to help policymakers answer questions like this one: how will elephant movements change if key fences are decommissioned? So in a nutshell, my project is taking real GPS data, plugging it into an agent-based simulation, and then trying to reliably predict savanna elephant movements under future changes, and specifically fencing in the region. But because I'm only 6 months into a 2-year postdoc, I'll focus this presentation on the simulation itself and its modeling inputs rather than validation techniques and final results. You can check with me. Maybe next year I'll present some of those. But first, I wanted to introduce the concept of an agent-based simulation. An agent, in this case, is just an individual elephant. We define a list of probabilistic rules for the individual's movements across the landscape based on real GPS data and landscape-level data. And then the model randomly chooses movement parameters within those rules that the simulated elephant will exhibit. These rules can be based on many features, but in particular, I'm going to be focusing first on responses to environmental features, activity states, and responses to barriers on the landscape, so those fences I mentioned before, and also roads. First, environmental responses. The following environmental relationships I'm going to present are already well established in this ecosystem, but I think it's important to quantify these relationships so that we can put them into the model correctly. So first, we know that elephants travel significantly further in the wet season than in the dry season. This is because an increase of rainfall brings with it an increase in available vegetation on the landscape. Here, I have 3 years of rainfall data shown. In blue is spikes of rainfall. You can see clearly the wet season versus the dry season where there's no rainfall. And then below here, I have the hourly distance traveled for these elephants, which I'm going to term step size during this presentation. And step size does not mean like the physical size of a step for the elephants. It's the distance in between hourly pings from a GPS recorder. So you can see that these step sizes really increase during the wet season. And we can quantify this using a linear mixed effects model with hourly distance traveled by season and then random effect of animal ID. And we have an increase of 115 meters during the wet season as opposed to the dry. We can do a kind of a similar analysis, but thinking about access to water. So elephants during the dry season really cluster around these water sources because they need to drink daily. But during the wet season, they can kind of spread out over the landscape. So this is GPS data from one female elephant over 4 years. And you can see that she travels way further from the Okavango River during the wet season. But I will also point out, you can see the fences are still restricting her movements. So again, we can look at a simple linear mixed effects model, looking at the distance from river as a function of season. We actually have an increase of 6,000 meters, or about 6 kilometers. Finally, we used a habitat selection model to investigate how elephant herds use different spatially distributed resources. In this case, is canopy cover class. So this is the Kasa region. In blue, you have more open landscapes, and then green is more closed landscapes. So very briefly, the way that this works is you have some kind of a landscape. Let's see, you can imagine this is one of those points on the map, and we take real data from a real elephant. We give it a bunch of available places that it could have walked to, and compare those to the ones that it actually did walk to. And if you compare those two sets of points, you can get a proportion in each cover class used versus available. In this case, it's going to be used as in black and available is in gray. The thing to note here is that in open landscapes, this middle bar, the proportion of used open landscape is greater than the proportion of available open landscape, meaning that these elephants are kind of choosing these over what you would expect from just random selection. We can also put this inside a generalized linear model with use as a response to cover class. And this is, I don't want to explain the full figure, but what I want you to notice is that the darker pink means that you're switching from one of the other cover classes to the cover class in the row. So in this case, elephants are choosing to move from closed or sparse landscapes into open landscapes. Okay, so next up we have activity states. So this is just the idea that elephants, when they're moving around the landscape, they're not just random particles, right? They are doing things while they are moving. And so we're going to use a hidden Markov model to decompose elephant movement into exploring, resting, and foraging states, which Gareth had mentioned in his talk on Tuesday, so thanks for priming the audience there. As an example, I'm showing you female elephant data from one elephant in 2015, and she's in Kaudum National Park in Namibia. So first, you might have a resting state. I'm realizing it's a little bit hard to see, but when an elephant is resting, you might expect them not to walk around the landscape too much. So taking short steps and having an even distribution of when they're choosing to turn from their last point. For any HMM nerds out there, I'm going to put up some of these graphs of frequency distributions of step lengths and turning angles, but you all don't have to worry about that too much. Next, you might have an elephant that's foraging. You might expect them to take a little bit longer steps and maybe turn around and take a little bit of a different approach to the landscape. And then finally, an elephant might be exploring or having a concerted walk towards a destination. And so with this female elephant, we can decompose her movement into these three pieces. And what I want you to notice is, maybe I can do this laser pointer, these central points where the elephant is moving out and then foraging around the landscape and moving back in, which is really cool, especially if you know anything about elephants. They love water. So these are actually water holes that they are moving out from and moving back in. All right, and the important thing with the activity monitoring is that, or modeling, is that you can take this and put it with the environmental pieces that I was showing before. For example, wet season release from river dependence. The wet season expansion pattern actually only seems to apply for foraging and resting states and does not apply for the exploring state. And this makes sense when you think about, well, these elephants are exploring to get water. So it makes sense that they're needing to drink still in the wet and the dry season. They're just foraging and resting further from water sources. Finally, we had to include individual responses to these different barriers on the landscape. There's a 2022 paper with Robin Naidu, who's one of my co-advisors, studying elephants in Botswana and Namibia. They found that linear features are differentially permeable to elephant crossing by feature type and by elephant sex. So here, what I want you to notice is that the bars for river and road crossings are higher than fence crossings, right? So it's easier to cross a road than a fence. And then also that the females in black bars are always lower than the males in gray bars. And again, this is because they're in larger groups. They might have young with them, and there's actually no bar for fence crossings, so the females are never crossing the fences. And here's an example of one of the females just in January of 2021. You can see she comes up across the fence and bounces off and goes along her way, but she's not able to, or chooses not to, cross the fence. And so once I had all three of these pieces together, I wanted to start modeling these and simulating these movements, but then I realized is that an agent-based modeling framework that did everything I wanted it to didn't really exist. So I had to kind of work on my own. So building on this great R package by Marshall and Duthie from 2022, I was able to add on to their work and add linear barriers onto the landscape and augment their code to take in actual environmental raster data instead of just matrices. For any wildlife veterinarians in the audience, you've probably done a postmortem of an elephant, but this is what a postmortem of an elephant for me looks like. This is a lot of the code that I've written for this project. And I don't put it up here just to show how much I've done, but also to say that elephants, again, are not just random particles on a landscape. So it takes a lot to make your simulation match elephant movements. So thank you. So in my remaining time, I'm going to go over how this model works. Sorry about that. So you start with some current position on the landscape. You choose an elephant's, randomly choose an elephant's activity state that might be in, resting, foraging, or exploring. And then you choose a landscape to match that. So when an elephant is foraging, they might be looking for different things on the landscape than when they're exploring. Then we choose, again, those random points on the landscape and add in all of our fencing data. And for some of these elephants that might, let's say this fence is permeable, the elephants might be able to cross some of the time. We're going to take that permeability and turn it into a probability of throwing out some of these points. So we throw out one of the points and redraw, and then have the elephant actually select a point on the landscape. So what this looks like for agent-based simulations, at first, hopefully you can see this elephant that starts out way down there. It just ignored the fences, and it took me a long time to get them to actually recognize the fences, but then I have some elephants wandering around the landscape and bouncing off the fences, and that was great. I was able to also add in some of these waterhole relationships that we saw before. So this is an ongoing process. I've just been doing this for 6 months, so again, I'm really excited to show more of this next year. And returning to a workflow from before, next steps is validation with real telemetry data. And I wanted to add in some other components I know you guys are going to ask me about, which would be density dependence, migration, and even some things that were mentioned in previous talks, like the species in memory and adaptive perceptions on the landscape. Finally, with my remaining time, just like stepping back for a sec, as the Southern African landscape warms and dries over the coming decades, pictures like this are going to be more and more frequent in news media, and thinking about these elephants on the landscape. Not suggesting that Hwangi's elephant death problem is related to fences because it's unfenced, but these kind of stories are just going to become more and more apparent on the landscape. So addressing this fencing issue when elephants need to move to access resources, especially during drought, is going to be more and more important. So with that, I'd like to thank everyone here and my funders and co-authors.
Thank you. To end off the session, we have Cornelius, who will be telling us about age structure and demographic inference in elephant populations.
First of all, Sandparks, thank you for the opportunity to present. My name is Hannes Low, and I am keeping myself busy with elephant population dynamics, demographic studies, and as most of you would probably know, is that demographic studies usually take many, many years to get sensible results. And even more so when you start to study elephants, because elephants, things happen rather slowly. So these populations of elephants can vary in their demographic responses across landscapes. To make sense of populations, you can either choose an easy to study population where population growth happens rather fast. The benefit of that being that whatever disruptions in age structure occur can be eliminated or diminished rather quickly, whereas with slow growing populations, those disruptions are not diminished that quickly. They last within age structures. So the point I want to make, which I probably failed to accentuate, is that we look at age structures. That's an alternative option to study populations of animals with a long generation life. That is what our studies entail. We look at age structures, and we choose smaller conservation areas where there's lots of surface water and where elephants are supposed to do fairly well. There's ample surface water. That's a key resource for elephants. And we don't expect in smaller insular conservation areas that there will be a lot going on. We expect these populations to grow rather quickly, in elephant terms at least. So the key to high population growth, of course, is survival probability across all age classes. And it is easy to imagine that things should be very similar in smaller conservation areas, but they are not. On the left you'll notice that there's a few populations that exceed 6% population growth, which we kind of think is actually above what is demographically possible for an elephant population. There are authors that claim they can grow to at 7% or so, or even higher, but we need to be mindful that there are actually a bit of confusion in literature. We need to be very precise in our definition of population growth. Is it a demographically driven population growth? Is it happening at a stable age structure? It is easy to have populations that grow at 6, 7, 8%, even higher, but not for a long duration, not for an elephant generation. That is simply not possible. So in these small conservation areas, some of them not so small, there's obviously also a few things happening. Everything is not that optimal. There are still droughts, and there's fire. There are disturbances which are related to management interventions, translocations, immunocontraceptions, and so forth. So trying to make sense of all this, one really have to ask yourself, demographically, how fast can an elephant population grow? And we have to go to the literature. There are not many studies that really give us the exact population parameters that will give us accurate population growth rates. But we essentially take what is available in literature. We go to a population matrix model, a Leslie model, and we simply project the population forward in time, and eventually, after 100 years or so of simulations, we arrive at some sort of estimate of what they are capable of. And that population estimate is what we call an asymptotic population growth. It's not a transient population growth where you get a 7 or a 8 or a 10% growth temporarily. This is asymptotic population growth. So there's differences, contradicting estimates in literature, but broadly speaking, without going in too much detail, an elephant, the first thing I need to mention is elephants are demographically buffered, meaning they have high survival probability, especially the adults. Where one would expect most variation to occur in elephant populations is amongst in the fecundity, the birth rates. So, age at first calving, about 12 years. Some studies have reported ages of 11, even 10 years. Surely that must be kind of an exception. And definitely not the rule. Age, a calving interval of 4 years seems about right. Again, studies that suggest slightly shorter calving intervals, but then again, a lot of studies, don't exactly report whether those shorter calving intervals are related to calf mortalities. Surely if a calf dies, a female will get into estrus sooner, and of course, the birth rate can reduce to perhaps as low as 3.2 years, but that's a calf mortality that caused that. So if we look at this, a reasonable maximum survival rate, I would say a 0.995 is reasonable. I mean, in some smaller protected areas, it could even be 1.0. No mortalities until the very late age, but that is probably a bit too optimistic. There should, in realistic terms, there should be some mortalities. So the most sensitive, asymptotic population growth is most sensitive to adult survival. And unfortunately, of course, adult survival is the most difficult to estimate. It takes a lifetime. You literally need a lifetime to get that sort of info. So it leaves us with the difficulty, if we want to answer the question how fast can elephant populations grow, we need more accurate information on adult survival. So just to put things into perspective, we have the Amboseli and elephant population that has been studied for decades. And if we implement in this Leslie model, Amboseli survival probability, you end up with a population growth of 4.5% or thereabouts. If you take survival probability under absolute optimal conditions and you put in that value of 995, then you are slightly above 5%. So we don't even get to 6%, really. Things have to be very, very, very favorable to get to that 6%. So yeah, in considering all this, we can't really predict how fast the population grows. We will be off by a 0.5% here or there. And adult survival is probably context dependent anyway. So this is what an age structure more or less look like. This is simulated, make no mistake. Real populations will never behave this well. You will have a bumpy road. There will be droughts and there will be slight disruptions that will mess up this pattern a little bit. Even so, if those disruptions are minor, then it's not a huge issue. The point is, we have a 50-50 split, give or take. If we induce, or in this imaginary exercise, we increase calf mortality a little, we can simply increase the calving interval. It has exactly the same effect. And then by reducing it by 20%, you end up with a 60% adult proportional abundance. So the point is that if in our simple model, if it is realistic, adult proportional abundance gives a very good, it serves as a very good indicator of population growth. So yeah, all I need to do then is go to the matrix and to change the calving interval and get a few points like I did there, and you get a very nice linear relationship between, and now you can choose immature proportions. I decided it just looks nicer. In immature proportions correlated with population growth, and there you are. We look at the bottom, that black line there. This is our simple model. This is what we think a population should, how it should perform. And now the question that remains, of course, is how do real populations relate to this nice relation, this correlation? And there's about 6 study areas. We notice immediately that Addo and Liwonde fits the line rather nicely. And they are well-established populations. Soronga is supposed to be a well-established population, and they are. But the thing is, the reason for Soronga to fit so poorly is that there was indications in reports that there was an influx of elephants to Soronga, so it boosted the population growth. The very important point here is that these 2 points, Addo and Liwonde, the demography and the actual counts kind of match up rather nicely. Pilanesberg and Madikwe, fairly recently established populations, and they suffer from a lot of founder effects, and you can't really match the population trajectory with your age structure data. And it makes things a bit difficult. Demographic inference is compromised. Waza Marsh, I just added it for the sake of interest. We have poor indication of what the actual growth is at Waza Marsh. This is just to illustrate, if you start with a population and you have biased sex ratios, it takes very many long years to remove that effect. So no wonder that populations and recently established populations have this high growth rate. So at the top, I induced a little bit of stochasticity in birth rates. That is fine. If I have a study area where you have that situation, you can take a snapshot of age structure and you can learn a lot about the population demography. Whereas at the bottom, if the population in harsher environments, if there's more stochasticity, then you're going to struggle. You need a lot of extra data. You need more snapshots. There's more variability, and a single snapshot simply would not do the trick. So I'm probably leaving out a few things, but never mind, my time is just about up. I would advocate for harsher environments. We need to collect more data on calf recruitment. That's where I think the most variation occur. And in these populations, these harsher, I suspect it's possible that populations growth it's much more transient, whereas in faster growing populations, it is much more deterministic. It makes interpretation so much easier. And my time is up. Any questions?
Thank you so much. We do have time for a few questions for any of the speakers from this session.
We were somewhat surprised at that our elephants are relatively unaffected in the Kruger. What are your thoughts about how long a drought should be or how extreme it should be before you start to see some influence on elephant demography?
Look, I mean, it is obviously, it has to link to the carrying capacity. And from theory, you know, elephant populations, relatively speaking, they get affected quite close to the carrying capacity, which we don't know exactly where that ceiling is. So it's difficult to pick up demographic responses, density-dependent demographic responses. It's difficult to demonstrate. I would love to learn more about, you know, the behavior of elephant populations in drier environments. It surprises me that data on calf recruitment is very sparse. Unless any of you have info, I would love to have a chat.
Any more questions?
Yes, Sam, I expected a question from Sam.
Thanks for various talks in this whole session and so forth. I think there are several concepts that we are throwing around that doesn't even make sense. Carrying capacity is one of them. You know, things are varying all the time. You've seen this lovely stuff from Sandra and so forth. But what was really fascinating to me is some of the work that Lawrence and them reported, and I was wondering, do we now need to design a rat culling strategy? and I'm asking this chicken tongue, but I think that these key drivers that are hidden somewhere, we are not thinking about it. So for example, can I predict the spatial intensity of use of rodents that are also varying over time and space, as Sandra has done for a whole bunch of other sort of things? And perhaps that can give us a bit of a better understanding why some elephants are bigger than others, or stronger, and why some trees are stronger than other places. Thank you.
I saw you said that you designed your study, or you decided on what areas you'd study based on their size, or the area size, so that it'd be simpler. But then when you went into explaining, there were a lot of variables that affected your populations, like drought. Some populations were established longer. So do you think, instead of choosing, like if you were to continue studying this, instead of choosing based on the area size, rather choosing parks that have had similar disturbances, so parks that have had droughts in the past for the same length of period, or those that haven't had any major disturbances, so that you can rule out those variables and just look at calf recruitment?
Yeah, I chose small parks just to kind of get across to set the scene. Small parks, size actually isn't that important. If it's a big area where there's lots of water supplementation, that should actually resort under the same sort of umbrella. Small parks, of course, have the problem that they quickly run out of space, and they are the problem areas. They don't have the buffering capacity to deal with high population densities. That's basically my simple answer to that.
I have two questions for you and for Ryan, so I'm not sure who would answer. But as I said, one question about the surveying method that we used in some of these parks, considering it's the 1990s in some cases. Did the methods change over time, and how do you think that would have influenced the population estimates? And then the second question was, with regards to population or percentage growth, you know, I always get, this is also a dilemma I often sit in with myself, but when numbers are low to start with, when they increase, the percentage growth looks higher, right? So if you think, if something goes from 1 to 1, it's 100% increase, but it's just one individual.
Exactly.
But
yeah.
from 10 to 11 is one individual, but now it's 10% increase.
Yeah.
So I wondered how you'd get around that. Thank you.
Yeah, look, I mean, that is the founder effect thing. Small parks, they are quite unnatural, actually. You know, I mean, it's exactly true what you say. If there's one male and 10 females, and he's fit enough, and none of them are pregnant, well, in 4 years' time, there's 20 elephants. And it's 100% growth in it. We know that's not demographically. We need to contextualize. We need to make it clear that demographically driven growth, it's different from just counts. It's fantastic if you can combine the two. If you have counts and you have demography and they match up, you learn a lot about elephant populations. If they don't match up because of founder effects, you need to literally know every individual personally, and from all that you make sense of the demography in the end.
Thank you so much. That brings the session to a close. Can we have a round of applause for all the speakers of the session? Thank you. Everyone can break for lunch.